osmosis, electrophysiology, and cell movement.
Okay, so first of all, I would like to thank the organizers for inviting me and giving me the opportunity to speak here. It's been a truly great conference for me. I've been learning a lot from the many great talks that have been given here. So my primary field is electrophysiology, and I am a more or less a complete novice when it comes to cell movement. But I have recently learned from many people, many people, including those in this audience, that electrophysiology and osmosis are very tightly linked to cell movement. And what I would like to talk about today is some basic electrophysiology, and also I'd also like to talk about some very recent attempts, my recent attempts to try to understand the interplay between electrophysiology and cell movement, and hopefully I can convey my sense that this is a truly fertile ground for mathematical modeling, and it is also exciting because it is at the intersection between two very classical and well-established fields within mathematical biology: electrophysiology and mechanics. So. So first of all, I would like to do maybe the first half of the talk is going to be a review of basic electrophysiology, and probably many of you here are familiar with electrophysiology, but the kinds of electrophysiology we have to talk about here is a little bit different from the kind of electrophysiology that you might be familiar with, and so hopefully there is something new for those of you who may know a bit of electrophysiology. And then I would like to talk about some of my recent attempts to try to talk about electrophysiology and its relation to cell movement. So let me begin with a discussion of some basics of electrophysiology. So the first thing that we all learn as students of electrophysiology is that the ionic composition inside and outside the cell are very, very different. So these are human numbers, representative human numbers. So the intercellular concentration of sodium is much smaller than the extracellular concentration of sodium, and the intercellular concentration of potassium is much higher than the extracellular concentration of potassium. So there's basically a 10 to, in this case, about 30-fold difference in concentration. And the chloride concentration varies between cell types, so we don't have to worry so much about this. There's also it's also very well known that the calcium concentration is very different inside and outside the cell. This and the chloride will not concern us. The main thing that will concern us is sodium and potassium. Now so in fact the fact that this ratio of sodium and potassium concentration, intracellular sodium and potassium concentration, that there is so much more potassium inside the cell than sodium, is actually a universal feature of life. So it's actually true for archaea, bacteria, and eukarya. So this is actually a universal feature of life, so this ionic composition has been used to it has even been used to try to get at some of the origins of life on Earth. But anyway, so one thing that that is very interesting about this is that if you let the cell alone, then you would expect that the sodium concentration and potassium concentrations inside and outside of the cell will equilibrate. I mean, without energy input, you cannot maintain this kind of concentration differential. And in fact, our body and our cells use a lot of energy in actually maintaining this concentration differential. In fact, your brain uses about more than 50% of its energy is used in maintaining this concentration differential. So what is the reason why cells have to do this? And there are actually quite a few reasons, but one of the reasons why this is a good idea is because the cell has to control its volume. So now I want to talk about why this concentration differential is linked to cell volume control. So the fundamental problem of cell volume control is the following. There are a lot of membrane impermeable solutes inside the cell, small organic molecules, nucleic acid fragments, and so on. And of course, so the cell is a biochemical machine. So you don't want these things to leak out through the membrane. So you have these small organic molecules inside the cell, but outside you do not, okay, to a very rough approximation. Therefore, the osmotic pressure inside the cell is always going to be higher than the outside, just because you have stuff inside that you don't have outside. And the cell membrane is permeable to water, which means that if you let the cell alone, then water is going to seep in, and eventually the cell is going to rupture. And the question is, and so the cell has to come up with some kind of strategy to control its cell volume. Depending on whether you are what kind of cell you're talking about, your strategy is different. If you are talking about a plant cell, the plant cell has a cell wall. So basically contains its turgor pressure by building a wall around it. I don't have the picture here, but if you are a prokaryote, then many of these prokaryotic cells, for example, paramecia, have these things called contractile vacuoles. So they are basically just water pumps. So they will pump water periodically to get rid of water that seeps in. Now, if you're an animal cell, then the control mechanism is fundamentally different. So animal cells control their volume by controlling their electrolyte content, their ionic concentration, and of course that sentence does not explain anything. So what I want to do is to try to explain how cell volume control is achieved in animal cells. So so this is a small table. So here, what I have is a table of concentration of proteins, positive and negative ions, inside and outside the cell. Okay. So let's say we have a cell, and the cell has 10. This is arbitrary units, and these numbers are completely arbitrary. 10 millimolar proteins. Okay. Proteins are proteins and nucleic acid. These are large molecules. So and so we're going to assume that each of these protein molecules has minus 10 charges. In general, many of the macromolecules that you find in macromolecules proteins are negatively charged on average. So we're going to assume that each protein contributes minus 10 charges. And these are, again, arbitrary numbers. Now, let's say we place this cell that has 10 millimolar proteins inside an environment that has 150 millimolar positive ions, 150 millimolar negative ions. So you can think of it as an NaCl solution, for example, if you want to. And we are going to assume that the valence of the positive and negative ions are +1 and -1 respectively, just for simplicity. Now the question we want to ask is, what is the amount of positive and negative ions we have to have inside the cell in order to get volume control? So how can we compute that? Well, we need, so if the positive ionic concentration inside the cell is x and the negative ionic concentration inside the cell is y, x plus y plus 10 is the osmotic pressure inside the cell. I have to multiply by, I guess, by RT, but up to a constant multiple, it is given by x plus y plus 10. Okay? And then this has to be equal to the osmotic pressure outside, which is 300. So x plus y plus 10 has to be 300 in order to have osmotic balance. Here I'm assuming that the membrane is too flimsy. It doesn't membrane or the even the actin cortex is too soft that it doesn't contribute any mechanical force. So the other thing I need is that Coulomb forces are very, very strong. So usually if you look anywhere in your body, it is electroneutral. So I'm going to assume that assume electroneutrality. I add up all the charges, it's equal to 0. So I have two equations and two unknowns, which I like to call so this is and you can solve for x and y. This is mathematical biology in action, and this is what I like to say. It's one of the simplest examples of mathematical biology that actually give you a non-trivial result, I think. And so what do we gain out of this? So what we gain is that the concentration of negative ions inside the cell has to be smaller than the concentration of negative ions outside the cell. The concentration of positive ions inside the cell has to be larger than the total concentration of positive ions outside the cell. And this basically comes from the fact that the proteins were negatively charged. So in order to maintain osmotic pressure balance or cell volume control, or in order to maintain cell volume, what you need is less negative ions inside and more positive ions inside. So the question then is, how can a cell do that? And the way the cell accomplishes this is by creating a membrane potential. So if you can create a membrane potential, that is let's say, so by convention, the outside of the cell is always 0 millivolts. I mean, and the intracellular side, the voltage inside is always measured with respect to the outside. So if the voltage inside is minus 70 millivolts, minus is negative in terms of voltage with respect to the outside, then positive ions would want to be inside, negative ions would want to be outside, and so there's a chance that you can get this kind of thing to work. So the next question is, how does the cell generate or maintain a negative membrane potential across the membrane? And this is done in the following way. So as we just saw, okay, the potassium concentration inside the cell is so much higher than the outside. Okay? And this is actually maintained actively. Potassium ions are actively pumped into the cell using chemical energy, that is ATP hydrolysis, and it is pumped into the cell. Sodium ions are pumped out, and this results in a large concentration differential, 30-fold in the table that I had at the very beginning. Now, on the membrane, there are what are called potassium channels. There are many kinds of ion channels, but for most cells in your body, the dominant ion channel is the potassium channel. So potassium channels are expressed everywhere in your body. Assume that the cell you're talking about only expresses potassium channels. Then what's going to happen? So what's going to happen is that because I have so much potassium inside and not so much potassium outside, and these and there are these potassium channels, the membrane, the lipid bilayer membrane is effectively impermeable to ions. Okay, this is basically soap film, and soap film doesn't have a lot of affinity with charges, and so ions are more or less cannot penetrate the membrane. So if you have these potassium channels, then the only ions that can go through the membrane are through the potassium channels, and potassium channels are selective for potassium, so it's only the potassium ions that can go through. Okay? Now, if that's the case, I have lots of potassium ions here. All of the ions except for potassium remain inside, and potassium will leak out a little bit. Now, as potassium leaks out, what happens is that the potassium will accumulate on the outer surface of the membrane as it leaks out. the lipid bilayer membrane acts as a capacitor. And after a while, so you will start to develop a membrane, you will start to develop a difference in voltage here and here because positive charges accumulate on the outer face of the membrane. And after a while, the diffusive tendency for potassium to go from the inside to outside because of the concentration difference, and the tendency for potassium to want to come back because now the inside of the cell is negatively charged, is going to balance out. And this is called this kind of generation of potential is called a diffusion potential, and the diffusion potential it's basically a balance between this kind of electrostatic forces with diffusion, and typically the voltages that are created by this process is on the order of minus 70 millivolts, or basically on the order of about 25 millivolts. I mean, so 25 millivolts times some constant. So in the case of our cells, the typical resting membrane voltage would be about minus 70 millivolts. So what do we learn? What have we learned so far? So the picture is the following. So in order to maintain osmotic balance, the cell must extrude negative ions from the cell and get more positive ions to be inside. And this comes from this simple calculation that we had. And this is accomplished by setting up a negative voltage inside the cell with respect to the outside. Now, how does a cell then create this negative voltage? Well, potassium is actively pumped into the cell so that the intercellular potassium concentration is high, and then there are potassium channels on the membrane, and that creates a diffusion potential about minus 70 millivolts. So this is how volume control is done in animal cells. Now so there are some interesting observations or questions, I guess, at this point already. First of all, I think probably many of you here are probably maybe familiar with action potential generation in neurons in the heart, and possibly the work of Hodgkin and Huxley, which was actually done here in Cambridge. So but that's actually an evolutionary afterthought. So the reason why we have a membrane voltage in the first place is because of, you can argue that it is because of volume control, and it is only later in evolutionary history that the existing membrane potential was then co-opted for neuronal signaling, for example, or cardiac signaling, for that matter. Now, another thing is that you see immediately is that ion regulation is tightly coupled to volume control. So then volume control is in some sense the simplest form of cellular shape changes. You can wonder whether ion regulation might be tightly integrated into cell movement, and the answer to that is a resounding yes. But before we go there, I would like to put a little bit more mathematics into this volume control business. So the standard way of modeling this volume control is what's called the pump-leak model, which I've a very simple version of which I have put up here. So this is following the discussion in Keener and Sneed's book on electrophysiology, and also of Peskin's book on mathematical biology. So I just have so this is just basically a balance of solutes inside and outside inside the cell. So first of all, we restrict our attention to sodium, potassium, and chloride, which are the major ions we find inside the cell, which are the major ions that contribute to osmosis, to osmotic pressure. So so let's talk about the balance of sodium. So V is the volume of the cell. So this is the total amount of sodium you have inside the cell. Na plus I is the concentration of sodium inside the cell. So what is the rate of change of that? The rate of change of that is given by two terms, one term here and the other term over here. The first term is the sodium that passes through sodium channels. Okay. So ion channels are passive. They don't use any energy. So what this is saying is that the sodium flux across the membrane is controlled by the difference in the electrochemical potential of sodium. Okay, this is what this is saying. Phi being the membrane potential, and Na plus E being the extracellular sodium concentration. So this is just the difference in electrochemical potential across the membrane, and that is driving the sodium flux across the membrane for these ion channels. Now, this minus 3 alpha, this is the flux that comes from the sodium potassium ATPase, and this is an active flux for which chemical energy, or ATP, is used. Here, for simplicity, I'm taking this to be just a constant, minus 3 alpha. And we also have the same kind of equation for potassium and for chloride. And for chloride, we don't have any active pumps in this model. Okay, so I don't actually think there are any chloride pumps. Maybe I'm wrong. So we have sodium, potassium, and chloride. We have the equation for volume. The volume equation is just that the transmembrane. So dV dt is equal to transmembrane water flow, and transmembrane water flow is controlled by the difference in osmotic pressure across the membrane. The osmotic pressure inside is given by the sum of sodium, potassium, and chloride plus the concentration of organic molecules you have inside the cell. So the organic molecules, the concentration of organic molecules you have inside the cell is total amount of organic molecules divided by the volume. And this A is constant, reflecting the fact that the membrane is impermeable to these organic solids. The important thing about this dV dt equation is that there is no such thing as a water pump. So water is, transmembrane water flow is completely passive. It only follows osmotic pressure gradients, and you don't have any plus something that is a water pump. And I think there is this general. So I think at present nobody knows any water pump, and so I think this is probably a universal feature that we don't have water pumps. Yes.
What about the issue of space? Because this is transport across the membrane.
Yes. Issue of space meaning I'm treating the cell as just one point. Is that what you mean?
Yes, just simply, yeah.
Yes, exactly. So this is yes, you're right. This is an ODE model. So we'll get to what happens if we want to do it in a spatially extended setting.
Because you have gradients, how would you?
Yes, exactly. So if I have gradients, then this is not going to be valid. You're right.
Yes. So far you assume that there's no aquaporins, just ion channels.
Actually, yeah. So you can assume that there are aquaporins. That the aquaporin, if there are aquaporins, this zeta would be a larger number. Yeah, that's a great point. So the lipid bilayer membrane itself is somewhat permeable to water, but if you have aquaporins, then it's going to be much more permeable to water. Yes. Yes.
What about the area of the cell? That's also changing.
Yeah. So the area of the cell. Yeah. So that's a good point. So So here I don't have any area of the cell here. So I guess if how would the area of the cell influence this? Like mechanical?
Well, if I have a gradient, then the per unit area flux is determined by the quantity in brackets, and I have to multiply that by the total area.
Yeah. Okay. So for these numbers here, GNA, GK, and GCL, so you would assume that the that these numbers are just proportional to the number of channels you have, so it won't probably depend on area at all. So if I assume that this zeta is only dependent on aquaporins, then the number of aquaporins will not depend on area either. If we don't have any aquaporins, yes, this zeta is going to depend on the area. And another thing I don't really know is whether these membranes are actually incompressible or not. I mean, we've heard some discussion about how membranes are folded, and as the cell expands, it just unfolds. In that case, zeta won't change either. Another thing I'm assuming here is that the actin cortex or the cell membrane contributes no mechanical force, which is again another approximation.
That's why the area goes in, right? I mean, because
yes, you can have the.
If you change the area, then you change the pressure balance. And
yes.
It goes into
Yes.
osmotic balance.
Exactly. So if I have that, then I will have to put that in, but I don't have that here. So I have 1, 2. So I have V and my sodium, potassium, and chloride are my unknowns, but I also have another unknown, which is my voltage phi, and that voltage phi is determined by this relationship over here, which says that the total number of charges I have inside the cell, Z is the average valence I have for each organic molecule. So this is the total amount of charge I have inside the cell is equal to transmembrane voltage times the capacitance of the membrane. So very often in the context of cell volume control, it is a very good idea. You usually take this to be equal to 0. You do a non-dimensionalization and you find out that effectively CM is very small. Yes.
I think your result critically depend on the value of Z. And can you give a rough estimate of the Z and a way to estimate it?
You mean this
Yeah.
Z?
Yes.
Yeah. I don't. Yeah, that's a good question. I think it's not very difficult to estimate what this Z is because people you can have this is a bulk property. You can have like cells and then fractionate the cells and really just look at the concentration of each ions and look at like charge balance.
Electrophoresis, you mean?
Yeah, basically charge. I mean, so you basically open well. So it's not necessarily so clear if I can get like precise numbers. Right?
No, I mean even a rough estimate because like it's clear that the Manning length and things like this.
Not really. I mean, this is actually a very macroscopic problem. We're not talking about things like Debye layers and so on. I mean, we're not there. I mean, we can talk about that too. I mean, that's an interesting topic. But this is not. This is a very macroscopic and kind of model.
So how much would you give for?
Yeah, like I don't know. That's a good question though. I don't know. Like I quoted the number -10 right in my table, and I think it's like minus something. I don't know.
Minus for sure.
Yeah, minus for sure. Yeah, -3, -4, -10. Yeah. That's a good question. I mean, I would also point out that this is probably a very crude. Okay, let's see. So the system. Okay, so the system for which this theory has been tested extensively is in red blood cells or red blood cell ghosts. Okay, in fact, which is red blood cells don't even have a nucleus, and if you make it into a ghost, that is basically you rupture the red blood cells and then you make it fuse back to itself. Then you can basically control everything that is inside the red blood cell, a ghost. And in that case, you probably know Z to a very good to some good precision. And there, the theory has been validated quite extensively, I think, since the 60s. But yeah, I actually don't know the exact numbers.
What if you have fixed charge density?
Yeah, exactly. So ZA is a fixed charge. ZA divided by V is a fixed charge density.
You could have also outside.
Yes, yeah, you could have also outside. That's right. Yeah. Okay, so let's see. Okay, so some of you may be familiar with electrophysiology a la circuit theory, and the connection is that you basically take this equation, okay, and take the derivative with respect to t, and then you get circuit theory. So usually, most of electrophysiology that people talk about in for example neuroscience is just talks about this equation more or less, and this equation is very good if you're looking at very short time scales over which concentrations don't change very much. But when you're talking about volume control, it's actually the time scales you're interested in are the time scales over which concentration change a lot. And so in fact it's actually we have to take another, so it's actually wise to take the other approximation, which is that Cm is equal to 0 and you have the electroneutrality condition.
Is that ODE well posed for C equal to 0?
Yes, that's a good question. So your question is, okay, when Cm is not equal to 0, so this phi is just so phi is just a function of sodium, potassium, and chloride and V, and you can just substitute it in, and you just get an ODE for V as sodium, potassium, and chloride. Now, if Cm is equal to 0, this is a differential algebraic system. Is it well posed? So the answer is yes, most of the time. Yeah, and you can actually write precise conditions under which this is a well posed problem. So the intuition behind this is that roughly speaking, the intuition behind this is that if you take this to be equal to 0, what happens is that phi evolves so that the electroneutrality condition is satisfied, and this is very basically analogous to things like the relationship between incompressible and compressible Navier-Stokes. So when you have compressible Navier-Stokes, you have so P as a pressure is a function of rho, whereas in the incompressible limit, you just have the divergence-free condition which determines the pressure. So here, if I let Cm to 0, then I get that the voltage evolves so that the electroneutrality constraint is satisfied. And okay, so the general structure of this model is the following. So you have this you have a balance of concentrations. I do not have to restrict myself to sodium, potassium, and chloride. I can have many more ions if I wanted to. And then I have the flux for the ion channels and the flux for the pumps. And I have the electroneutrality condition, and I have the dV/dt equation, which is that the volume changes with transmembrane water flow. So in general, this is a differential algebraic system with V, C1 through Cn, and phi as the unknowns, n plus 1 differential equation, and 1 algebraic constraint. And there was this question of whether it's well posed, and it usually is. Actually, whether it's well posed depends on some properties of this Jk, but it usually is okay. In some sense, this is the simplest model wherein electrophysiology and cell movement, so to speak. I mean, if you think that volume changes in volume is cell movement, if you do think that way, then this is the simplest model in which electrophysiology and cell movement is coupled. So this model is called the pump-leak model, and it's been around for a long time. So this was introduced in the early 60s, and Tossison and Hoffman were primarily interested in volume control, and their experimental system was erythrocytes, red blood cells. And this model has been extended and modified by many people, and it is really widely used in investigating water and solid transport, especially in epithelial systems. Since we're in a math institute, I might as well talk a little bit about math. So there are some analytical issues that you might wonder about, which is okay. Let's say you're given an equation like this. Okay, I am claiming that this is a system of equations that describes homeostatic volume control. So you would like to say that it has a steady state, maybe just one if possible, and that that steady state is stable. So if you can prove that kind of thing, that would be interesting. And you indeed can. I mean, you need some. Of course, you need some conditions on Jk and Pk. So these are functions of phi and c. under if you place some restrictive conditions, you can say a lot, and if you place some very weak conditions, you can nonetheless say something about it. And basically, if JK and PK do not behave too wildly, you can actually show that steady states exist, and that steady states are stable. And the key, and I'm not going to go into how to prove these assertions. The key to this is this identity, and I bring this up because it's beautiful. I'm biased, of course, but because it's also useful. So the thing is that you compute this quantity g, and g is basically as you can see immediately, it's basically the free energy of the system. And the free energy of the system, if you compute the time derivative of the free energy of the system, you get the difference in chemical potential across the membrane times transmembrane chemical ion flux, the difference in water potential across the membrane, or the osmotic pressure across the membrane times transmembrane water flux. So this is a kind of a canonical relationship that you see in any book on non-equilibrium thermodynamics. And so what I want to emphasize is that this model that I just presented, this Pump-Leak model, although it looks kind of complicated with algebraic constraints and so on, is actually has a thermodynamic structure to it. And if you exploit that thermodynamic structure, you can actually prove certain analytical results on existence of steady states and stability. For us, what is important is that this energy identity is going to serve as a guide in obtaining PD generalizations of the model. So there was a question from Anatita as to whether this point model or this ODE model was valid. Well, if we're going to be talking about cell movement, then it's not going to be valid. We'll have to talk about gradients of concentrations and so on. So we'll have to generalize that ODE model to the PDE setting, and this energy identity is going to serve as a guiding principle as to how to do that. Now, before I move on, I would like to just talk about this surprising fact that I learned embarrassingly very recently. So the membrane potential. So I said that the membrane potential is negative, and it's about -70 usually, but it really varies from cell to cell. Okay. And the surprising thing is that the resting membrane potential seems to correlate very well with how differentiated a cell is. So if you look at glia, neuron, and skeletal muscle, neurons and skeletal muscle, you always think of them as never dividing. Maybe they do divide sometimes. I don't know. But neurons, skeletal muscle, cardiac muscle, they don't divide. They're basically terminally differentiated. They have a very negative voltage. Now, even among differentiated cells, liver cells are known to divide. I mean, if you cut the liver, then it's going to grow to its original size. And they have a membrane voltage that is higher. If you look at tumor cells, you have a membrane voltage that is higher. And then, and if you look at the fertilized egg, a 4-cell embryo and 16-cell embryo, the fertilized egg has the lowest, smallest membrane potential. And I don't think, as far as I know, I don't think anybody knows why. So it's not. Yes.
All these channels can be regulated by.
Yes.
They can be turned off.
Yes, of course, yeah. So yeah, in that sense, yes. I mean, of course, you can go in and check the channel expression, and then that will tell you that the membrane potential is negative or whatever. But the question is, maybe the question I'm asking is, why does this have to be so? I mean, is there any kind of advantage to having a lower, smaller membrane potential for non-differentiated cells compared to having very low membrane potentials for differentiated cells? Why this seems to be a universal property, I think nobody knows. I mean, whether there's any kind of advantage to having physiological advantage to having this kind of arrangement. It is also the case that the membrane potential changes with cell cycle. So as you see here, if you going from G1 to S, there is hyperpolarization, meaning that the membrane voltage actually goes down, and then as you go from G2 to M, there's membrane depolarization, meaning that the membrane potential goes from say -70 to up. And again, these things are, as far as I know, people don't really understand why. I mean, it's been known since the 70s that this is the case, but certainly there's no mathematical modeling as to why, or to any of this. And so, and this is not exactly maybe cell movement, but I think I just want to throw it out as a very interesting snippet of information that hints at some very interesting interplay between electrophysiology and the things many of us here care about. So now I want to come to the question of the link between electrophysiology and cell movement. So ion channels, water channels, so aquaporins, ion transporters are implicated in cell movement, and this is unequivocal in the sense that you, for example, you block certain types of channels, potassium channels, the sodium hydrogen exchanger, for example, and so on. In some cell types, it drastically inhibits cell movement, so in this review paper by Albrecht Schwab, he proposes as one aspect, I guess, of this crawling motion that has been the subject of many a talk here. So you can think of this as regulated volume increase in the front and regulated volume decrease in the rear. I do not think that I would personally think that the actomyosin contraction mechanism that has been talked about by many people here is the primary mechanism. But after all, 70% of the cell is water. So you need some plumbing to in concert with the actomyosin mechanism to get the cell to move forward, and that plumbing is probably achieved by what is here and maybe at the rear. And it is indeed true that many of these channels that I have here, or transporters that are depicted here, localize to the leading edge. So an extreme example of how osmosis and electrophysiology might be related to cell movement was proposed by a group from Johns Hopkins last year, their experimental results are the following. So you have these cancer cell lines. So this is I think it was a sarcoma cell line and breast cancer cell line. And they put them in a confined 1D environment, and they move at 20 microns an hour. Okay, so this is relatively slow, but it's still moving, and it does not depend on actomyosin and microtubule systems in the sense that the movement is not inhibited by latrunculin, blebistatin, or nocortisol, and the movement is inhibited by sodium hydrogen antiporter blocker and aquaporin. So this is A, aquaporin knockdown. That's roughly speaking their primary finding, and so the question is how does this work? So what they propose as a working mechanism is the following. So if you have this cell over here, and somehow what's happening is that you have solutes up here, and the cell is actively pumping solute from here to the inside, from the inside to the outside. The solutes are being pumped in this direction. Now, if the solutes are being pumped in this direction, you'll have a higher concentration of solute here rather than here, or here rather than here. And if you have a higher solute concentration here rather than the outside, then water is going to flow from the extracellular side to the intracellular side, and then from the intracellular side to the extracellular side. And so as the water moves through like this, then the cell will have to move upwards. So this is there.
Is the water stationary, the outside water? Is it flowing?
Yeah. So that's yeah. So the water is stationary. That's what they say. The water is stationary, and and you have this and so the transmembrane water flow directly. So if you're moving with the cell, yes, the water is moving through you. But if you're looking at the lab frame, the water is stationary, and the cell is moving upwards.
and the water cannot go. I guess the cell is completely attached to the boundary side, that there's no water that's going to flow.
Yeah, that's what they think. That's their experimental setup. The cell is basically completely obliterating this channel.
But you need some adhesion, otherwise you wouldn't move anywhere.
Yeah. So they have some. Yeah. So the cells are adhered. I mean, they have some kind of. Yeah.
You have attach and detach, otherwise
Yeah,
you just
I think.
the
Yeah.
average should be.
That's right. It is detaching and attaching. I think that's what they think. So it's basically just crawling through. Yes.
This still leaves open the question, if you go back to your previous slide, whether cross-membrane pumping has any role at all.
I'm sorry?
This doesn't really mean cross-membrane pumping has a role to play in the normal crawling of a cell.
Oh yeah, this we don't know. Yeah, we have no idea. So this is So there's no this is no proof that for normal crawling this is relevant. You're absolutely right.
Yeah. That's actually an interesting question.
That's true. Yeah, that's right. I completely agree.
No cells can pump fluid from a basal to an apical surface or apical to basal. And so you would expect in those cells, if you confine them in a slippery tube, that they would move because the
exactly. I completely agree. So although and this is exactly what you're saying. And although the authors of this paper, the osmotic engine paper, did not say this. So this is not new. I mean, this is kind of old. So it's been hypothesized since the 60s that how and generally accepted, I think, that water flow through epithelial systems is basically done in this way. So in the basolateral membrane you have sodium potassium ATPases, and solute is actively pumped out into the extracellular fluid, and you create a difference in osmotic pressure, and that creates water flow, and that is how water is absorbed in our intestines or in our kidneys. So, and in some sense, the osmotic engine mechanism is just this, but it's not. But water is not the one that's moving. The cell is moving instead. So the and if you compute the absorption speed, it's actually comparable to the osmotic engine mechanism. So it might also give some partial support to their idea that this is indeed a osmotic mechanism.
Sorry.
Yes.
Is the pumping of the water only at the front end, and why?
It's by the way, it's not the pumping of the water; it's pumping of solute.
Yeah. But is it always at the front end?
No, it's not necessarily. I don't think. Well, so what they feel that it's so the people who wrote, so the Johns Hopkins people and the people so Shansun, whom I'm working with. Oh, it's dead now. Okay. So I think that most of the action is at the leading edge, but that's not necessary. I mean, I think it's okay. I don't know why that should be true. I mean, it's okay if the pumps are at the back rather than the front, I think. Yeah, and indeed in this epithelial system, sodium potassium ATPases are basically here. So it's in some sense it's at the back end rather than the front. Okay, so the question that Jimmy raised was whether is this even relevant to this motion, and of course we don't know, and so in order to even try to address that question, at least if you are doing some computation, then we have to come up with a model, and we have to come up with maybe at least two-dimensional model to actually try to model this whole process and try to see whether something can be done. And I'm just going to talk about some very preliminary attempts on my part to actually try to do this. So so we have different types of ions. So the different types of ions are going to be satisfy this electrodiffusion equation with phi being the voltage, and then we have some advection. And that advection comes from the equations, hydrodynamic equations. Here I'm assuming Darcy plus Darcy law plus Stokes law. So that's so some people might call this the Brinkman equation. And then the voltage is determined by the electron neutrality constraint. So, and we have incompressibility. Okay. So that's the equation that is satisfied inside and outside the cell. But of course, I mean, the interesting thing is what happened to the boundary, because especially when we're talking about osmotic pressure and so on, everything is happening at the boundary. That's what's interesting about all this, I guess. So so this equation is kind of standard. Okay. So so we have large X, which is the position of the membrane. Okay. So theta is the material coordinate, and x theta t is the position of this membrane. So I'm writing everything in 2D, which may not be just for okay. So we should do this in 3D, but we're currently only doing this in 2D. So the boundary condition for concentration is this here. This is just a conservation relation, and the only thing that is a little bit different from the usual conservation relation is that you have to take into account the fact that the velocity of the membrane, so the cell membrane itself is moving. Okay. And more importantly, the local fluid velocity u and the speed of the membrane is different. And this is the important thing about this, right? So because there is transmembrane water flow, because there is transmembrane water flow, for example, because there is transmembrane water flow, the speed of the local speed of the flow, okay, and the speed with which the membrane is moving is different. And the difference between the two is actually the transmembrane flow. So if u and dx dt, that is if the underlying fluid velocity and the speed of the membrane were the same, then, for example, by incompressibility, the volume inside would not change. And that is not what we want, because if water comes in, the cell is going to expand and so on. So the very kind of maybe one of the most probably interesting things about this fluid structure interaction problem is that there is a difference between so we don't have the no slip condition for the membrane interface.
The membrane is a non-material surface for you.
Exactly, that's right. It's not a material surface for you. That's right. That's right. It's its own material surface, and it is moving not with the underlying fluid velocity, but it has a difference, JW. Okay. Now so the rest of it is really simple. So you have force balance, and the force balance is that the difference in stress across the membrane is equal to the elastic force exerted by the membrane. Here I am not neglecting membrane elastic forces or acting cortical stresses maybe, and so that's the equation. So and this JW JW, which is the transmembrane water flux, will depend on. It doesn't say here actually. Okay. So it will actually be proportional to this guy, but I'll come to that right now. So okay. And one of the the interesting thing or the important thing about the equations that I just wrote down is that it satisfies this energy identity, and in that sense, it is a kind of a genuine generalization of the pump-leak model to the PDE setting. So this gives us confidence that we're not really missing out on any important physics. And another thing that we get out of this is that what the transmembrane water flux should be proportional to. So this expression tells us the transmembrane water flux has to be proportional to the difference in osmotic pressure, but also the difference, also the difference in pressure as well. So it's going to be the sum of the difference in pressure and the sum of the difference in osmotic pressure that is going to control water flux through the membrane. And it's not really the pressure itself, but it's the normal component of the stress, which makes sense if you think about it. Okay, so at present we are not able to do simulations, full simulations with electrostatics, and so what I'm going to show you now is just when we have one solute that is not charged, in which case we don't have to do electrostatics, just the mechanics, but we also we do have to solve the concentration equations, and if you look at this, the only place where energy is pumped into this model is through this P. Okay, so it's actually, so this is a pump, so there's some pumping mechanism on the membrane, and all the rest of the equations here is just passive. There's nothing active about this. It's just the pumping of the solute. Okay, that's the only place where energy is being input into the system. Everything else is passive. So we developed a numerical scheme in 2 dimensions to get to do this. We have a uniform Cartesian mesh, and so for the fluid structure interaction problem, we use the immerse boundary method because it's simple. The really difficult thing is how to solve for the concentrations, and the reason why it's difficult is roughly speaking is because of the following. So the concentration field cannot be smoothed. So you see. One thing that one likes to do often in these interface computations is to use level set methods, phase field methods, and so on that allow you to somehow, or immerse boundary for that matter, that allows you to somehow smear quantities inside and outside so you can treat it as just one function. Okay? Now, the problem with that approach in this particular problem is the following. So you see, so let's say I have let's say I plot concentration as a function of x, and this is, here it's inside, here it's outside. The concentration actually has a jump. So the sodium concentration, for example, the potassium concentration, for example, is going to be high inside and low outside, for example. Now, although here I'm only thinking about one solute, so it potentially has a jump. Okay. Now, okay, so if I use some kind of smearing method or level set method, then I will be thinking about a function like this. Now, the problem with doing this, okay, is that if we do this, we won't be able to enforce boundary conditions. And the reason why we won't be able to enforce boundary conditions is probably clear if you look at what they look like. So the boundary conditions look like d grad c dot n is equal to c times something. Now, if you look at this smeared out thing and this thing over here, well, the values may be comparable, maybe sometimes, but the derivatives are completely different. So it's not. So as far as I know, it's very difficult to do any kind of smearing or regularization to produce just one function throughout the domain to get these things to work. And therefore, we really have to work hard and do some computational geometry on the membrane to produce reliable results. But we have been kind of partially successful. So it was a lot of hard work. And and so what we do is one thing so let's say the position of the membrane was here first, and then it moves to a different location. Then as it moves to a different location, you have to, so reflecting the fact that the membrane has moved, sometimes you have to prepare initial values on grid points that have newly become intracellular or extracellular. And that's a necessary step that one has to take. And once you've prepared those past values, then you can solve the advection diffusion equation. And then once you have that, you solve the for the velocity field and so on. So it's a moving boundary diffusion, as far as I can tell, is kind of a is a difficult set of PDEs to solve all this. And you can and under certain kinds of parameter values, you can get the 2D cell to actually go forward. And these, by the way, these values are still completely arbitrary, and they don't correspond to any kind of physiological value. So I don't know whether this is really true or not, but at least in principle, it's possible. So what we have here is that at the front we have pumps that are pumping solute in, and at the back we have solutes that are pumping solutes out. And so let me see. Okay, so there we are. So what I have here is so this is one simulation that we've performed. So this is so it actually okay. So it's starting from an ellipse, and the and it doesn't show, but the at the beginning the concentration field is completely uniform, okay. And the membrane has some elasticity and some bending stiffness. So you have pumps at the front and pumps at the rear, and it is pumping solute from the inside to outside. What is being plotted is the concentration of the single solute that we are simulating, and this solute concentration gets depleted in the front of the cell and gets accumulated in the leading edge of the cell because the solutes are being pumped in. Now, because of that, you create a concentration gradient across the semi-permeable membrane, and so water flows in.
So is your cell continuously increasing in size?
That's a great question. Probably, I don't think so. Initially, it might be increasing in size, but the elastic membrane and the bending rigidity also has some amount of, has the effect of containing volume. Yes.
Sure, I probably missed it. What are the rules of interaction with outside world? How does the cell interact with surface or matrix around?
Oh yeah. Okay, that's a great question. So that's a good question. There's a Darcy term or a Brinkman term. Which means that implicitly there is a matrix, and then there's a friction force between the underlying matrix and this cell. So it's just velocity times constant.
But which velocity?
The velocity of the fluid.
Of fluid
inside
the cell.
Inside the cell, yeah.
How can there be interaction of the fluid inside the cell with the surface?
Yeah, that's a good question. So it's so this is kind of a yeah. So that's a so I guess yeah, that's a good question. So I think the I mean you can imagine, I guess, that that maybe the velocity inside has a parabolic profile, so that it has some friction force with the bottom. But you're right. I mean, this is really just a proof of principle type of calculation, showing that it is possible. And another thing that is very interesting in regard to that is that let's say we take the Brinkman term and let it be 0. Let's say we have this zeta u, or the Darcy term, zeta u, and let zeta to be equal to 0. The cell actually moves, but very, very slowly. What happens is that the cell is more or less stationary, and it just creates a flow, and doesn't move very much. It just kind of it moves very, very slowly. It's only when you introduce some friction with the outside world that you can actually move. So yeah. So you can also have.
There's a quick question.
Yes.
Can I just check what you said then? Are you saying that it's got a higher concentration of chemicals because it's sucking it in from the outside?
Yes.
So, but the chemical field around it,
Yeah.
towards the end of the simulation.
Oh, sure, yeah.
zero.
So what's happening is, yeah. So what's happening is that we have pumps that is pumping out here and pumping in here.
Yeah.
So these pumps here are pumping out,
Yeah.
and the pumps here are pumping in.
But what is it pumping? 'Cause it looks like it's zero around the front of the cell.
It's not. Yeah. Well, yeah. It's very close to. Yeah. It's very close. I guess it's. Yeah. It could be very close to zero because the pumping is very strong.
So it's sort of like instantaneous. That it's maintaining the high level inside because it's just sucking everything in.
Yeah. Yeah. Yes.
How are the pumps distributed around?
Yeah. So the pumps. Yeah. The pumps are. Yeah. Here in this example. In this example, the pumps are here and here.
Okay.
Yeah. And here the pumps are going out, and here the pumps are going in. Yes.
Have you got a pressure gradient?
Yeah, there is a pressure gradient too. We haven't. I don't have a picture to show you the pressure gradient, but we're computing that too. So it would be interesting. I mean, this is not. You're right. I mean, the water transmembrane water flow is not just controlled by the osmotic pressure, but also by the mechanical pressure. It's the sum of the two that is controlling the water flow. So this concentration profile is not completely indicative of the force or the water or the difference in water potential that is driving the flow. Yes.
Seem to get a shape that's very. We get this shape very often with completely different models.
Right. Right.
Propelling the cell.
Yeah.
Does that shape depend on the distribution?
Oh yes, it does. Yeah. It depends a lot, and we don't know what it depends on at all. But I can tell you, this is a different. Let's see if I can get it to play. This was maybe this was the same. This is the same. No, this is not the same. Okay. So this is a different simulation where the pumps were actually distributed more widely at the front, and then you get a different shape. So yes, it will depend on many parameters, and we don't really know at this point. It will also depend very much on the rigidity of the membrane. If we make the membrane extremely rigid, then it's just going to translate. For example, just as a circle, we have simulations like that too, circle like that. Yes.
Does your geometry reach a steady state? Or?
That's a great question. So we think so, but we don't know. There's a problem with the simulation actually. So going into the ugly details of what's happening. So it's not entirely clear if it's because of the simulations or because of the model. Okay. So it may turn out that the model is just ill-posed. So for the following reason. So basically what's happening is that we can't. So in these simulations, at a certain point, sometimes we cannot do the computations any further, and the reason is that the membranes seem to develop kinks after a while, and this may have to do with the fact. So basically, you see, for example, the cell may look like this, but may be developing some kind of kinks very close to some places, so that that is leading to some kind of that leads to some kind of numerical instability, and we cannot go forward. We do not know whether this is actually a numerical instability or whether it's actually a problem with the model.
I thought you said you had bending though. Doesn't that regularize?
Yeah, actually, that's right. So we put in bending to regularize these things, and actually, when the bending force is strong enough, it's okay. So in that regard, maybe it really is some kind of thing like that. But it's also possible that it's a numerical instability. We don't know. Yes.
One question which to connect this to the things that people have been talking about before.
Right.
Is even if you don't think that water pumping per se is the primary means of motility, if you have these substantial calcium and potassium ion gradients, are those sufficient that they're going to be modulating your actin polymerization and your myosin activity.
Yeah, that I think is.
You really need to be including these gradients in the models that we build of,
I think
actin and myosin.
I think that's possible. I also
We're more thinking about calcium as a possible regulator.
Calcium is of course very important. Although calcium are in such low concentrations that they probably wouldn't affect osmotic pressure. But you're right. And but I would think that maybe another thing that is I think important about this is, let's say you have a lamellipodium that is kind of moving through. You can assume that you have two options. I mean, if the membrane could be completely impermeable to water, then water will have to basically go around the lamellipodium, basically for the lamellipodium or the cell to go forward. The other option is that the water just goes through. Maybe that's actually better or more efficient for the cell. For the cell, the water just goes through the cell rather than goes around the cell, and maybe this is and there are a lot of aquaporin, for example, knockout or aquaporin inhibition experiments wherein you actually see that the cell movement is arrested when you introduce these things, and it's very possible that yes, the actin-myosin machinery has to be working to get these things through, but 70% of the cell is water. You need some kind of plumbing so that water will actually go through as the actin-myosin mechanism does its thing. And in order to really capture that, you would need to be able to do these computations where water is actually flowing through the cell. So that's that. I wish I could show you some real simulations, but those were really, in some sense, very toy models that would allow me to probably get published in computational physics journal, but nowhere else. And but hopefully we are making some progress. Finally the computation was done by Ling Xing, who was a postdoc with me and who's now in Case Western Reserve. He really did the heroic work of doing all the computational geometry to get this to work. The initial model coupling solute and fluid flow I developed with Bob Eisenberg and Chun Liu in a paper that we published together. I've been very fortunate to be working with Sean Sun and his postdoc since last year on this problem and some other things. And I would also like to thank Alex and Leah for introducing me to Sean in a workshop in Banff on cell motility last year that allowed me to get in touch with them and work with them. Okay, so thank you very much.
Some questions? Just one in the original Skinner-Snide model there.
Yes.
The chemical potential given by a logarithm of the concentration.
Yes.
In the spatial model you presented, that it was without the logarithm. So would it play any difference?
I don't think it will make very much of a difference. We could do the logarithm as well, but I don't think it will make a lot of difference. That's a good question though. Yeah.
Other question would be, what would happen if the pumps wouldn't be distributed in such a orchestrated way? Let's say it would be more randomized, or if there would be just, let's say, the inlets located on one end and the outlets on the other.
It would be different. I don't know. So if it's randomly distributed, as long as the membrane tension is strong enough, I would think that it will still be able to maintain its shape in some way. one interesting thing you can think about is what happens if you have pumps only on the front and not much in the back. We've done simulations like that. It does seem to get the cell to go forward, but what happens is that the cell at the back forms cusp-like singularities very quickly, and we can't really do the simulations further. So there's an interesting question of how these pump distributions lead to different shapes and so on. Yes?
In trying to assess this mechanism with an engineering mindset,
yes.
So what are the maximum fluxes, these JWs, that are accomplishable realistically in a system?
That's a great question. I don't. So that's a great question. I think that can be answered, at least in the biological context, by looking at epithelial water transport. So these are basically systems in the intestine and kidney where the system is maximally optimized so that the water will go in as quickly as possible. So if you do the measurements here, and I think these are classical measurements that everybody does, if you get the numbers here, that will tell you what the largest numbers you can expect are, I think.
What question here?
This all presumes that somehow the pumps get polarized.
Yes, exactly.
You're already starting with them polarized, right?
Yes.
So something's got to polarize them, and I'm guessing that the actin side.
Yes, absolutely. Yes. So that's.
Their answer was in that paper as to how they might get polarized.
Yes. So exactly. So they think that the authors of this paper think that the actin cytoskeleton is very much involved in getting for example, the sodium hydrogen exchangers onto this side rather than this side at the very beginning, and they also they also have this hypertonic shock experiment and hypertonic shock experiment where here they have a hypertonic shock here and the cell reverses direction. This is actually a very, very puzzling phenomenon that they think they purport to explain, but I don't really understand. Anyway, so they have this experiment. They have a hypertonic shock, and the cell goes in the other direction. If you block the actin-myosin system at this point, then this response is abolished. So what that means is that so the sodium hydrogen exchangers and aquaporins do not localize to this end unless you have the actin-myosin system. Right.
Maybe a naive question, but why are you running your simulation on a free cell, not in a confined cell like?
We can do confined geometry, but that would be a 1D simulation, and it's easy to do. It's easier to do in some
But you have no indication that a real cell would behave like this carrot.
Yeah, that's true. That's true. Yeah. So maybe I should say it this way. So in this one-dimensional geometry, you can write a simple model that allows you to. So in this one-dimensional geometry, you can write down a simple model that is in 1D, and you can simulate it. And this had been partially done by them in this paper, although I think there are still many improvements to be made. They have done that, and so the question we wanted to start asking was basically to come up with some kind of a general framework that will allow us to assess whether these things are really important, not just 1D confined geometry, which is very restrictive, but something that is a little bit more general.
But you have no proof from cells that they would behave like this.
Yes, we don't have any.
Take the cells and destroy the actin.
Yeah.
I guess they will not move.
I completely agree. So we don't have any proof that it will move in this way. It's basically the point is that we're trying to build some kind of computational apparatus that would eventually allow us to assess the role of fluid flow in concert with actin-myosin contraction. So we'll have to in fact overlay this kind of model with an actin-myosin type model to actually understand the interplay between the two. What we have here is just the osmotic part.
But how do you want to prove that your model is right?
That's a great question. I don't know. I don't know. In fact, I don't even know whether they have been able to prove that their model is right. So this is just 1D, and what their model so this is just 1D. Okay. So let's see. So right, and their model consists in doing a very simple PD model of just of a concentration of just one solute. Now I do electrophysiology, and it is highly deficient if you're working with just one solute. I mean, you at least need to have sodium, potassium, and chloride, and their explanation, at least to me, their model explanation of why the cell reverses under hypotonic shock is not very believable to me, at least. And so even for this thing, I think it's very challenging to try to see whether their model is correct, because a lot of the channel parameters and so on are very difficult to measure.
But
Yes. Yeah. So I don't know. Yes.
Just to follow up that question, there are cells that move without any actin. I mean, if you look at the sperm cell of the nematode, it has a completely different system. It doesn't have any actin. It moves perfectly well. And there's been some modeling in that suggests that there's a pH gradient or something inside the cell that somehow drives.
I think Alex had a model.
Yeah, Alex and where is Alex?
Yeah, but there's a major sperm protein there which has property very similar to actin.
Okay.
So pH gradient is very relevant there, but it just kind of establishes polarization at the front and disassembly at the back. But there are definitely cells moving completely without actin, like some nerve cells. But then they based on microtubules. So this is another example. Just flow drives the cell. What puzzles me is what's the coupling between the inside of the cell and the outside.
I see. Yeah.
Flow cannot do anything about it.
Yeah. It's I feel that the experimental results are really very interesting, but I think my feeling even about their paper is that there's a lot to be done in really understanding what is really going on. I don't think, although their model, they claim that their model explains their data, it's very, at least for me, it's very difficult to understand. For example, these hypertonic and hypotonic shock experiments are very difficult for me to understand.
I think one last question.
I was just curious about the membrane voltage and differentiation in the cell cycle. Do you know if anybody has looked at the connection between those two? Because I know the cell cycle you get stuck in G1 right before differentiation.
Yeah. So I can give you references. I don't know. I think what I learned very recently is that there were extensive studies done by a physiologist named Cohn in the 70s that really that described the phenomenology. And so it's been known since the 70s, but it's not.
Have there been any models of?
I'm not even sure if there are any models. There seem to be verbal descriptions and verbal theories as to why this might be happening by Cohn himself.
I've seen models relating cell cycle length and differentiation to CDKs and cyclins. And
Right.
It actually worked quite well. So it's just
exactly. So this may be another layer, but it's striking that you have this kind of universal variable that correlates with differentiation. Maybe this is because of my lack of knowledge, but I really don't know any number that is universal to every cell that correlates with differentiation. I think it's very interesting.
Okay. Thanks. We can go for lunch.